So this is the culmination of my master's project that I did here at Bristol, and I'm now a PhD student at Virginia Tech. Uh-oh. It's not working. Oh wait, I got it. That's fine. So as long as people have found fossils, they've been recreating what extinct animals look like. And you can see here, these are some early reconstructions, and we know now that ammonites didn't actually fly. We know that sauropods didn't live in water because they were too heavy to live on land. But this is a good representation of what we had over a century ago when we had very limited fossil evidence. However, certain things have taken a while to catch up as well, because this is what dinosaurs looked like when I was a kid. And you can see that they're portrayed still as these large, lumbering lizards. They're brown and green. And I always remember hearing when I was growing up that we would never actually know anything more than this. This was the best it was going to get. The fossil record only preserved so much, and there were certain things that would never be preserved. So we would never actually know what these animals looked like when they were alive. However, if you asked a kid today to describe a dinosaur, chances are they would talk about something a lot more like this, because we know now from the fossil record that dinosaurs had feathers. And there are extraordinary implications for studying feathers from the fossil record, because we know from looking at modern birds that you can tell so much about their biology, their behavior, sexual dimorphism, and the environments that they lived in by studying their feathers. Sinosauropteryx was the first fossil dinosaur to be found with feathers, and you can actually trace that downy layer of filament down his back and up his tail. And in 2010, researchers dubbed Sinosauropteryx the first ginger dinosaur. And they did this by looking at melanosomes. Melanosomes are the organelles that contain melanin, and by looking at modern feathers, we know that there's a correlation between the shape and size of the melanosome and the color of the feather. So at the top in the black band, those are eumelanosomes, and they're found in black and dark gray or dark brown feathers. And at the bottom in the red band are phaeomelanosomes, and those are found in red feathers. And the moldic impressions that were found in Sinosauropteryx's tail were very similar in morphology to phaeomelanosomes. However, as recently as 2014, people have questioned whether or not melanosomes are actually preserved in the fossil record, and people have suggested that they're indeed bacteria. So, for my master's, the first question we decided to ask, which is kind of obvious question, is can we detect melanin, traces of the chemical melanin, in fossils in association with these structures? And we weren't the first people to ask this question. In 2012, Johan Lindgren and colleagues looked at the dark spot of an Eocene fossil fish eye, which contained these structures, and they looked at it using mass spectroscopy. They compared the spectra of the fossil fish eye to a eumelanin standard and picked out 55 characteristic chemical peaks that matched between the two. Then in 2014, they followed up with this study, and they looked at marine vertebrates, and they found similar 55 chemical peaks, which they compared to a eumelanin standard. And they also tried to quantify this data by using principal component analysis. So they measured these 55 peaks, measured the areas, and put it into a PCA. However, all they were able to show was that they plotted distinctly. So you see that the taxa plot together, but there is no real connection between the fossil samples and the modern samples. So now comes my part of the study. I used time-of-flight secondary ion mass spectrometry, which works by bombarding the surface of a sample with primary ions. Secondary ions are desorbed and pulsed into a flight tube, and then the time it takes for the fragment to get to the detector is how it's measured. The lightest fragments get there first, hence time of flight. And TOFSIMS is a fantastic method for analyzing fossils because it's minimally destructive. If you have a small enough fossil, you can just put it straight into the machine. You won't even see that you did any analysis on it because it only uses such a small amount of material to get an entire chemical spectrum. You can also do in situ analysis, which was great for what we were doing because you can actually see a distinction between the feather and the matrix that it was in. And unlike other types of mass spectrometry, it does not average the chemical information, so you actually get a spatial distribution on the sample surface. So these are some of our results. These are the spectra from a eumelanin standard, a fossil frog, and the matrix that that frog was in. And you can see again, there's a qualitative similarity between the eumelanin and the frog, and then the matrix looks a bit different. This is one output that you get from TOF-SIMS. The other you get is this chemical map. So you can see here, this is a meso feather that we analyzed, and each box represents the same area on the fossil, but it shows a different intensity of each of the molecules. So you can see that some of them are present only in the feather, some of them are present only in the sediment, and then some of them are ubiquitous across the sample. So we showed here that the characteristic modern melanin fingerprint is preserved in fossils by comparing these chemical spectra. But we wanted to take it a little bit further. So we decided to ask, how does this chemical signal change in deep time? Oh, that got a little messed up, sorry about that. So the way we did this was by doing thermal maturation experiments. And we took 9 species of bird feather. We saved the part of them to do unaltered fresh samples. And then we took 2 samples each at 200 degrees and 250 degrees Celsius and wrapped them in gold, welded them shut, and put them in an autoclave for 24 hours. And this is a sort of accelerated diagenetic process to kind of speed up the process and see how it works. And this is based on a 2012 paper by Maria McNamara, where she looked at how the morphology of the melanosomes change when you subject them to these sorts of environments. And we found that melanin degradation can be predicted using these methods. So here's my PCA. Again, I took those 55 characteristic peaks, measured the area, and then plotted them. So here are the fresh samples, here are the fossil samples, and here are the negative samples. So like in the Lindgren paper, you see them plotting distinctly from one another. However, when you bring in the maturation samples, you have 200 degrees, and then you have 250 degrees. So the matured melanin actually fills in this gap between the fresh and the fossil samples. So while we weren't able to replicate the changes in chemistry exactly, it was a good approximation for what was going on. So melanin degrades in a particular way, and that signal is preserved in fossils. And then this is a diagram that shows you the fresh and the fossil samples and the colors that we inferred based on our morphological and chemical data. So the trace box are the fresh samples, and then you can see the fossil samples go from eumelanin to phaeomelanin. So using a combination of chemical and morphological data allowed for us to make these inferences of ancient color. So, conclusions. Chemical traces of melanin are preserved in direct relation to putative fossil melanosomes. Melanin degradation can be approximated using artificial maturation experiments, and changes in melanin chemistry and melanosome morphology are correlated and show distinct patterns in modern and fossil samples. So I would like to thank these people, particularly the Palaeontological Society, for providing the travel fund for me to be here today. Thank you.